Okay, so this is the last lecture for Psych 3280, Neural-Based Subconsciousness. And today I'm going to talk about frontiers in consciousness research. Okay, so just to start, to recap the week 11, in week 11, I outlined the integrated information theory of consciousness, which starts with a proposal of the level of consciousness, and then there is a boundary of consciousness, and then within that, how integrated information composes, determines the contents of consciousness for the humans. And I introduced it quickly, how to compute the integrated information, and also the complex, which corresponds to the border of consciousness, in using a very simple situation model. And then I also, towards the end, explained how IIT can explain consciousness in patients without cerebellum, why there is no effect on the consciousness, and also split brain. Why split brain patients seems to have two consciousnesses within one body or one skull, and then conjoined twins who can potentially have two consciousnesses, and then with a shared input or thoughts possibly. All right, so this week I'm going to outline the frontiers in consciousness research, and with some exemplary research from my own lab. And then also lists empirical questions with strategies and frameworks and techniques that can be addressed. And then also situate the consciousness research in a broader context to wrap up the course as it's at the conclusion. The learning objectives of this week is to be able to answer the questions such as what does IIT propose about the Qualia? This is a sort of a last missing part of the IIT. And then how can we characterize Qualia structures? So the structures are the key concept in this week's lecture. And then I also will address the question of what are the possible empirical experiments that can be used to reveal the relationship between the Qualia and the brain structures. And then finally, what are the societal impacts of consciousness research? And then I added my recent paper with Dr. Alex Meyer, and which outlines some of the future perspective of the consciousness research as an optional reading. Okay, so just to get started, I start from the question from week 11, but actually this has been the sort of the remaining question for over the last 5-6 weeks actually, which is that how does IIT explain contents of consciousness? And for short, I will use the term Qualia. Basically, what Qualia means is the contents of what you are seeing, like redness of the red, or shape of the object, or face, whose identity, or whatever you are conscious of. That's what I call Qualia. And IIT explains level of consciousness as a sort of big phi, or the set of the integrated information that is lost at the level of a system. How does it actually get collapsed by cutting by introducing the minimum information cuts? And the border is corresponds to complex in IIT, which is a local maximum of the subset of the physical substrates that maximize this big phi. And then the Qualia, or contents of consciousness, corresponds to a connected pattern of the small phis. So each of the components can generate integrated information, as you if you recall from the last week. And then how does it actually get connected and then form the pattern that corresponds to exactly what you are experiencing at each moment? That's what IIT tries to explain or proposes. And first, I'm going to talk about this, Qualia or structure according to IIT. So the fundamental, just to remind you, the fundamental questions in consciousness research has been that, the correspondence between what you consciously see, and what your brain is doing. And, in particular, a subset of the neurons, neuronal activity. How does it correspond to, qualia, basic qualia of the dog? And this link between mind and matters is the fundamental questions in consciousness research. And some people call it as a hard problem. Some people call it as different names. And we have lots of clues about the link. This is the theme that I've been trying to establish over the last, weeks. So one of the example I have emphasized is this example of the brain area around, ventral surface of this, human brain. And here we have a cluster of the areas which becomes activated when the stimulus includes some kind of face. And then you perceive that, oh, there was a face. And also with, associated identity, emotion, and also gender and so on. They tend to elicit activity on both of the sides, actually. But the more important thing is that the right side of the brain tends to have a cluster which can also generate face percept when it's activated. So these lines are the ones that corresponds to the pairs of the electrodes. When they get stimulated, they elicit the face percept. On the other hand, next to that, there is a cluster of the areas that prefers color stimulus. Okay, and also when it's paired and then stimulated, they produces a rainbow-like experience. And of course, as a subject who are implanted in this electrodes, they have no idea which one is selected and then stimulated each time. So this is the best probably we can get to in terms of the causal explanation between the qualia and the brain activation. Okay, so but the key for us is that it's not really exactly where this kind of thing happens or the existence of something like this happens. The critical question is why stimulating these areas elicit the qualia of color and face. Because if you don't know anything about the brain areas, then they look really homogeneous. And if you use a microscope and then to look at the details of the neurons that are there, or if you use a DNA analysis, most likely you don't find anything different between this red part and the blue part. But somehow there is something that goes on within this red areas and the blue areas that eventually gives rise to difference in quality of the experience of the face versus color. What is it? That's the question. So just to summarize the clues from previous research, fusiform face area, FFA, V4V8, or middle temporal lobe, the neural activity in these areas are strongly, first of all, correlated with and also necessary for, and can elicit the qualia for face, motion, and the color. Okay, and necessity is established through the specific bilateral lesions of these areas, and then people have reported, loss of face, loss of color, or loss of motion. That sort of suggests that there is something in these areas that generate or corresponds to the qualia of face, motion, and color. And importantly, when stimulated, each of the areas seems to generate these, qualia. And why? That's the question. So to address this question, what I have been proposing, and also many other people also have similar kind of idea, is that we need to understand these kind of situation in the following ways. One is that what is the structure of the qualia? And then another one is what is the causal structures? And then finally, to understand the relationship between these structures. And this week's lecture is dedicated to some extent on what it is that what I mean by each of the structures. Okay, and the first one, Qualia structures, corresponds to, let's say, in this case of listening to some kind of sound, gives rise to experience, and then that experience itself, the relationship or similarity or inclusion or something like that gives rise to this shape of the sound or structure of the sound. And by this, just briefly, in the case of the sound of a C is similar to D or B in the tone. However, when you go further, different kind of tones, C, D, E, F, and then A, and so on, then one octave above C starts to sound similar. So if you encode the similarity of the sound, then the sound structure has been proposed to look like, helix like this one. C becomes similar to this one. So here the similarity is encoded in terms of distance in this structure. And a different kind of qualia has different structures. That's a sort of the idea here. So what is the causality structures? That's the next topic. So the brain, just to remind you, is a huge network of neurons, and it's really huge, humongously huge, and we can't really have any idea of what kind of structure it is in reality. And the number of the neurons in our head is roughly 10 to the 11 neurons. And in the current Earth, we have roughly 10 to 10 people, 8 billion people. So it's 10 times more neurons in your head than the people on the Earth. That's the level of this gigantic structure in the brain. Okay. And by the way, I might ask you these numbers for this this week's questions. It's pretty easy, so just remember this. Okay, and also this corresponds to and each of the neuron is connected with roughly 10,000 to 10,000 or maybe sometimes 100,000 neurons, other neurons through synapses. Okay, and the key here is that each of this connection is not to directly passing some kind of message or information. Okay, unlike people, each of the neurons doesn't speak to each other. What it does is just to change the probability or the likelihood or tendency for the other neuron to become more likely to be excited or less likely to be excited in the next time step. Okay, so excitation or inhibition of the connection through of the interaction between one neuron to the other, that is the essence of this neural network. And that when it's properly configured, it can compute something, and then that result in many other behaviors or sensory induction or transduction of the signals and things like that. And there is something going on inside the brain of this causality structures or causality network that seems to give rise to this face or motion or color qualia. And in terms of the integrated information theory, this corresponds to what it has been talking as information. But information in IIT is always intrinsic information. And to unpack this a little bit, this is difficult, so you don't need to remember, and I wouldn't ask it in the quiz, but it is the way the network causally constrains its past and the future. If you remember this, figure from the last week, to compute integrated information, the first step of this integrated information started with going from the current state of these neuron pairs, and then how this neuron pairs on off state transits into the next future of on, and also where it came from in the previous set. So these are all defined by this, modulation of the excitation and inhibition through the synaptic connections between neuron A and B. And that corresponds to this causality structures. In this case, it's extremely simple, but you can imagine that, each of the neurons is connected with thousands of other neurons, and each of the neurons has a unique, way to connect to other neurons. Sometimes it's neighbor, sometimes in a long range, and that patterns of the causality has a huge, possibility. But it seems that our brain is having this type of the connectivity in a preferential way, in a specific way, in each of the brain areas, and that most likely corresponds to, or at least underlies with our specific qualia. Okay, so then what I mean by this content of consciousness. So this is an again a very simple recap of the integrated information theory. Initially, we have this repertoire or patterns that this system can take, and then we compute this how much it constrains itself, intrinsic information, and then how much we lose by if we cut the system minimally. That's the integrated information, small files here. Okay. And then the important thing to realize is that you can do this kind of operation, not only the neuron between A and B, but if there is a neurons C, then you can compute that between A and B, A and C, B and C, and also A, B, C as a whole. So you can extend this kind of analysis on all of the subsets of the things that are composed with complex. By the way, the complex has to be determined first to get which one is boundary of consciousness, right? And then within this boundary of consciousness, all of the possible subset has to be computed and then constitutes the patterns of the causality structures according to IIT. And this is very, very difficult to do in practice to compute, but that's the sort of theoretical prediction. So in the real brain, this is difficult to do, or at least we cannot do that at all in the current technology or current our knowledge. But what we can do is to do the simulation. So Hong and Tononi last year published this paper where they connected these 8 neurons, A, B, C, D, E, F, G, H, in this way. And then each of these neurons are neighborhood, between the each, like C is connected to both B and D, and the D is connected to C and D, and so on. It constitutes this one line of this network. And each of these neuron is also, excites itself. And then when you set this, weighting parameter or excitation inhibition parameter to some extent, then you can actually compute this integrated information for these sets. And even just 8 neurons, remember we have 10 to the 8 neurons in the brain, the number of the possible ways to consider the subset is really humongous. So this part is the all the possible combinations of A, B, C, D, E, F, G, H, one at a time. And then the next one, this is all the pairs, A, B, B, C, C, D, D, E, and so on, but only those pairs that have a non-zero integrated information. And in this case, let's say, H and E is not connected directly at all. So in the next time step, it doesn't influence its future. And one step before, their E and H state doesn't do anything in the jointly on either E or H or both EH, and therefore they do not give rise to any kind of phi. So that's a phi of 0. So here, the two neuron pair only happens for the this simulated network only with the neighbors, and then this is all 3 neuron network that has a non-zero 5, and then 4, 5, 6, and so on. Okay, and then this is already super complex, but what they did was to also reveal how they are connected or how they are related to each other, and that's impressively, visualized by this super complex figures. And you don't need to understand this, and I wouldn't ask this in the quiz. But what they did was to visualize the sort of the magnitude of this small phi, or how this small integrated information is related by the some kind of encoding of the magnitude and how it's related to each other. Okay, so let's say, for example, ABCD here is related or closely located to ABC, or CDE, or BCDE, because it's kind of similar to each other, connected, and then it's far from other things like D, E, F, G, and so on. So this embeds the sort of the structure of the causality among this 8 neuron system, and you don't need to understand this complex things. And in general, this week's topic is highly advanced, so you don't need to understand everything I'm going to say. Okay, so in the simulated brain, we can compute the integrated information to some extent, but how can we actually make a progress? So what one of your tutors, Angus, has been working on is to apply this type of the analysis on recording obtained from the flies. So the very small Drosophila flies actually have neurons inside of their brain, and the number of the neurons they have is 10 to the 5 or something like that, and 10 to the 5 to 10 to the 6. So it's much smaller compared to the human brain, but it's again super complicated. Each of the neurons is connected with others, and the structure of this brain is, globally speaking, it's kind of similar to the mammal brain like structures, how the sensory information is connected and collected into the central brain, and then it's it's used for the future control of motor behavior and so on. And what we did was to use these signals called local field potential. It's similar to EEG in the human brain, and then try to generate this integrated information structure out of this. And this is just a sample kind of integrated information structure that Angus got. So here, this is a sort of the we can only compute integrated information structure or causality structures up to recording from 4 electrodes. And so what we did here is that, let's say, naming it as A, B, C, D, we computed how these the 4 channels constitute this integrated information structure. These yellow dots are the ones that are the contribution to this entire big phi, entire system level integrated information by each of these electrodes, and then green ones are the ones that are the pairs of these neurons or channels, and then the blue ones are trios. Let's say this one is D and B and C. So this is a DBC channel correspondent. And the purple one is the ABCD channels. And skipping all these details, what we found is that in general, the recording from wakeful flies are much more bigger in terms of structure, in terms of the magnitude, and also more complex compared to the anesthetized flies. So that's in a sense consistent with what IIT has been predicting about the biological system, and so this is something nice that we kind of working on these days. And previously, we also worked on whether this kind of thing can be also computed for the humans, and it's much more larger scale kind of recordings, intracranial recording compared to the flies local field potential, but still there we are able to get the perceptual report from subjects and then computed integrated information structure. And there, what we did was to show some kind of stimulus such as face or blank or something like that, and then we masked it with input to the other eye. This is a continuous flash suppression situation, and then subject reported whether it was a face or not, and then rated the visibility. And then we then focused on these recording areas of the face area, as which is supposedly we didn't do that, but if we were to stimulate these areas, that would probably have elicited face percept in these epilepsy patients. And then we did the analysis, and then looking at all the possible subsets of the 4 channels, 2 channels, and 3 channels, and so on, like Angus did. And here is the results of this paper. So what I'm showing here is 3 different structures that we computed. The left side is this face percept that was elicited during the backward masking experiment. So here we showed some stimulus, face stimulus, and then masked it with a noise stimuli just after that. And then here is a similar kind of experiment, but using the continuous flash suppression. And then the right side is the case where we made the face invisible by the continuous flash suppression. So the right 2 sides are the cases where we ask the subject to do the same task, and then the left side is completely different task. But in terms of the quality of the experience, these left 2 sides correspond to seeing a face, and the right side corresponds to not seeing the face. And then what we found here is that structure follows basically the percept rather than the tasks or stimulus input itself. Okay, so we are actively working on this kind of experiment and also data analysis to see the relationship between the causal structures and the qualia, and that can be investigated empirically. So the question now we can move is that causality structures related to qualia, and then are they unique in FFA and V4, V8 and MT to generate all these face, color, and motion qualia. The idea is that if we were able to compute these causality structures at the level of the possibly single neurons or maybe population of neurons, then maybe there is some kind of a level of the analysis which really uniquely determines the shape of this integrated information structure or causality structures that are unique to face on one hand, and then unique to color, and then unique to motion, and all these structures or shapes are similar across all the people who can experience all these things. And once we get this structure, maybe we can also reverse infer that when you observe this shape in this particular area, then this person must be experiencing motion or color or face. And that could be done both in a wake state or a dream state or drug hallucinatory state, or with or without reports, or with or without attention. It doesn't matter. That it could be the direct relationship between the causality structures to the qualia. That's the hypothesis, but we don't know whether it's going to be true or not. And then to address these kind of questions, again, this is a super advanced topic, but I'm not personally doing this type of experiment, but the recording technique has been improving really massively over the last decades really. If you remember that by the time that consciousness research actually started, it was around this time, 1990s. And there, we had human neuroimaging started, and then started to see monkey binocular rivalry experiments and all these exciting kind of time. However, as you will see that after that, the number of the neurons that people were able to record at the same time, which is extremely important to measure this causality structure, steadily increased in a log axis. Initially it was like only one neuron at the time, where we can have no idea about causality structures, up to 10, and then 100. That's the sort of 2010. And then recently, this neuropixel probes has been invented, and then now we can actually record up to 1,000 neurons. And this is a brain of the mouse, where these neuropixel probes were penetrated in the brain in many areas. And in Monash, we also do this kind of recording in mice and rats and also monkeys. And then what it can do is that with a single electrode here, they can record 70 neurons in the visual cortex, 60 neurons in the hippocampus, and thalamus, 200 neurons at the same time. And these analysis, these data recording technique, together with the computational analysis development, will allow us to get to the sort of the causality structure analysis into the future. And that's a pretty hopeful kind of blueprint into the future. So in summary, this first part, which was quite advanced, and so you don't need to understand everything I said, but just look at the key figures and also remember the key numbers, like number of the neurons and number of the synapses. Okay, integrated information can be computed in a hierarchical manner. So from A, B, C, we can compute A, B, A, C, and C, A. And that's a sort of a hierarchical manner or composable manner that I'm talking about. And then we can estimate such a structure of the neural network in principle. And then the hypothesis is that, or at least it's not really a hypothesis. Okay, so when there is this specific excitation and inhibition and the connectivity structures, then this causality structure should depend on the specific neural connectivity. And this causality structures has to be specific within an area as well as across the areas, to the extent that neural connectivity patterns are different between these areas. And these are most likely to be the key to explain why faces, colors and emotions are likely to be generated in different areas of the brain, and also at the same time experienced in us at the same time. And then the question is that can causality structures explain a different qualia? And that's the topic I'm going to explain in the next part.